Okay, so the first speaker is Vincent Loret. And I did not prepare any. I assumed that everybody knew everybody. For me, Vincent he loves methodologies and he's also a clinician. How is that? And he wants things to happen, evidence to be good, because then he has to see the patient. So that's the role that Vincent has.
I also think that we should give Rosa, Anna, or Celia, the organizers, a huge hand, because to assemble all of this together for an incredible opportunity to think, to advance the field. It's always wonderful to be able to talk in person, exchange ideas, and move the field forward. So thank all of you. Thank you for invitation. And is there not a better venue than Firenze? And we meld the arts and the sciences together, right? So I'm Vin Loret. I'm at the University of Pennsylvania, and my charge was to provide you with recommendations and practices in validation studies and their uses. So just as means of an overview, I think this audience probably knows why we do validation, but just to reiterate, what is the rationale for validation of algorithms for health conditions? So I'm talking health states, health outcomes of interest in real world data. I'll then segue to talk about what are the current questions and challenges in conducting validation studies. And then our group recently published a core concepts in pharmacoepi on validation. So I'll talk about the conventional approach to validating case finding algorithms. And then I'll conclude with use of the validation indices, the measures, the parameters that we get to inform quantitative bias analysis, which will be a nice lead-in for Dr. Schultz. And then I'll conclude with the use of artificial intelligence to identify health conditions, which Dr. Lippi will be talking about later. So I think it's evident for this group, but just to reiterate that case-finding algorithms can identify health conditions in real-world data, but let's acknowledge that those algorithms can be inaccurate. When I refer to an algorithm, I'm referring to an operational definition of a health condition in real-world data. And by health condition, that could be a condition to identify the study population. It could be the condition to identify the exposure. It could be the outcome of interest, the covariates, any of those. Using an individual diagnostic code, some group of codes, some combinations of codes, diagnoses, and other data. And it's analogous to a diagnostic test. We're trying to use the algorithm to classify the presence or the absence of the condition. But like any diagnostic test, those algorithms could be inaccurate. They may not perfectly classify the condition. So we can have people without a disease that the algorithm can classify as having the disease. That's a false positive error. Alternatively, we can have people with the disease that the algorithm classifies as not having the disease. That's a false negative error. And given all of these inaccuracies, we conduct validation studies to understand the degree of misclassification with these algorithms. And a validation study really is an approach to assess the accuracy of the algorithm compared to some reference standard, some data source expected to classify the individuals for the condition better than the algorithm. I don't refer to it as a gold standard because typically in healthcare data, it's very rare to have a perfect gold standard. So I use the term reference standard. And these validation studies will yield indices, parameters, measures. I use them synonymously. Sensitivity, specificity, positive predictive value, negative predictive value, which are valuable to help us inform the degree of algorithm error and help us to understand misclassification. And there's different approaches to these validations. In the most extreme conservative cases, we could determine the condition in all persons. That's often not feasible, typically not feasible. In some instances, investigators may say, we're going to use the algorithm to screen for individuals, but then we're going to determine the condition in all cases identified by the algorithm. That happens from occasion to occasion. But most commonly, what I'm going to focus on, and what often is done in pharmacoepidemiology, is we determine the performance of the algorithm in a sample of individuals. And that's really what I'm going to focus on for the entirety of this talk. In my role in pharmacoepidemiology, I am often confronted with questions, challenges with validation studies. Most commonly, the first question is, Vin, is it really worth the cost and the time to do validation? Which variables should we validate? Can I just use an algorithm that was validated in another data source? Hey, I'm going to use a US data source, but they validated this algorithm in Taiwanese data source. Can I just use that for this real world data source? How do I determine the case definition of a health condition used in practice? Which data should be used for the algorithm for the condition? Which indices for validation should I target? What threshold is fit for purpose? What sampling approach should be used? How big should the sample be? And how do I use algorithm indices to inform my main study? These are all the host of questions and challenges with validation studies that I am often asked. And I'll try to touch on some of these questions throughout. So I want to pivot to what is the conventional approach to algorithm validation that is typically used in pharmacoepi today. So often investigators will start with selecting a health condition of interest. And that may seem evident, but there is some thought that investigators often should give to this. There is a difference between selecting a health condition with a chronic indolent condition versus acute severe condition. And one must give thought to the health condition that will potentially minimize misclassification. So I study lots of liver diseases. So I'm going to use a liver disease example. If I wanted to study cirrhosis of the liver, that is a clinically silent condition that may not present for care. Very challenging to ascertain the timing and the presence of that event. In contrast, it would be, for me, more advantageous to study an acute severe condition like decompensated cirrhosis, end-stage liver disease. These are clinically overt complications. Variceal bleed, the person is vomiting blood. Ascites, their belly fills up with fluid. Encephalopathy, they're mentally confused. These are not clinically silent conditions. These are overt. And so those are individuals that typically will present for medical care, and it's often easier to identify. And so considering that is often at least an initial important step. Usually, once you've determined what the condition of interest is going to be, then really the next step is to formulate what should be the case definition that is used often in clinical practice. This requires investigators to go back and review the clinical literature and to ensure that you are collaborating with clinicians, patients, data experts, perhaps biostatisticians, all sitting around the table to best understand how might we formulate this case definition. I want to go back to the example of decompensated cirrhosis. What we did in our validation study, we identified 4 conditions that classify the presence of decompensated cirrhosis: ascites, peritonitis, variceal bleed, encephalopathy. We came up with formal definitions, and those who would be classified with the outcome if they met any definition for any of those diagnoses. And I'm actually simplifying this even more, because if you go back to the paper, we had a definite diagnosis, a possible diagnosis, or no diagnosis. And we will talk about that in this and other talks as time goes on of the value of that. So once you've formulated the case definition, you've devised the case, you next have to devise the case finding algorithm. Typically in pharmacoepidemiology, we use structured data. That may be medical diagnoses most commonly, but it also may be procedures, drugs, labs, some combination. But as we are increasingly coming into machine learning, AI, we in the field are increasingly using unstructured data like clinician notes, radiographic reports, pathology diagnoses, and pathology reports to enhance our ability to identify health conditions. Lots of options for case finding algorithms to identify health conditions. You could use diagnoses alone, but if you use diagnoses in the hospital, one must be aware that in some data sources, there is a principal or primary diagnosis. There are secondary contributory diagnoses. So you have to think, what do you want to use? What if you want to use outpatient diagnoses? Maybe you could use those. Often people use 2 or more outpatient diagnoses because they don't want to use diagnoses that are suspected but not confirmed. Lab abnormalities may be used. I study drug-induced liver injury. Clinicians typically use liver-related laboratory abnormalities at outcomes. Some will couple both labs and diagnoses or diagnoses and drugs. Lots of options to consider. And this is valuable when you sit down at the table with your clinicians, with your statisticians, with your database experts to identify what are the data and the elements that you have in your real world data. So once you've identified the case definition, the case finding algorithm, then the next step is really to determine what should be our reference standard. And in some data sources, it may be a disease registry or patient or clinician questionnaires. So we've collaborated, for example, with the Clinical Practice Research Data Link, where we've given questionnaires to general practitioners who have served as the gold standard. In many other instances, the medical record, the chart, serves as the gold standard. And investigators will extract components and data from the chart and use that as the gold standard. Again, it depends on the data that you have and the access that you have. Then the next step is to really prioritize what should be the validity measures of interest. Selecting the sample and then collecting the data necessary to classify that condition. Ideally, we would love to maximize all of the validity measures, but in reality, as Rosa had alluded to, we're often making trade-offs. And those trade-offs and that prioritization of the algorithm validity measures will depend on what the goal of the algorithm is and perhaps what your main accuracy concern is. So often what we do in pharmacoepidemiology is we're comparing drug A to drug B for some health condition. And so often we're looking at the risk ratio of this condition in drug A versus drug B. Often the main concern might be false positives and the focus might be on PPV and specificity. But Rose also alluded to sometimes we're interested in incidence, absolute risk, or perhaps risk difference of conditions with drugs. And so the concern may be false negatives. And in those instances, we may be interested in high sensitivity. Alternatively, if we're interested in identifying the cohort in which we will study, identifying patients for condition for eligibility, the focus may be on false positives, PPV, and specificity. So we need to consider what is the prioritization. We then select patients. If we're seeking PPV, we may take sample who met the algorithm, NPV, those who did not meet the algorithm. If we're seeking sensitivity, we may have a sample of true cases against which to test the algorithm. For specificity, a sample of true non-cases. But I highlight in red, this is usually irrespective of the exposure group, and that's something that I'm going to come on to. Often when considering sample size, we generate that based on the expected estimate of the parameter, maybe an accuracy of 80% for a particular condition, with a width of the confidence interval of plus or minus 10%. And then if we're using medical records, we will select components, data in those components to abstract, some time window of review, and we may wish to decouple the abstraction from the adjudication in which to blind the adjudicators to the presence of a drug. And training and piloting is an important part of that. And then we get to the adjudication of the condition. And that could be as simple as agreement with a disease registry or a questionnaire, or it could encompass expert review, either of the charts itself or often more commonly structured forms to confirm the condition. Usually the adjudication encompasses one or more clinical experts who will review the data forms using the formulated definition from step 2 and adjudicate is the condition present or absent? And as I mentioned earlier, it may be, do they have a definite diagnosis, a possible or probable diagnosis, no diagnosis, or there isn't enough data to make the assessment. But it is a standardized, reproducible process done to reduce intra and inter-rater error. And we often assess adjudication procedures after 5 to 10 chart reviews to ensure the fidelity of the procedure. We typically will use 2 clinical experts to assess the replicability of the process, often measuring the kappa statistic. And if there is disagreement between the reviewers, employ a third adjudicator to arbitrate the case and serve as a tiebreaker. And then lastly, we determine the algorithm accuracy. Those are the validity measures. Most often, it's PPV. Often, if the PPV is over 80%, investigators consider that fit for purpose. One of the things I think we're going to discuss in this meeting is that true, and what does that mean? And if the PPV is less than 80, people consider to refine or to refine and then revalidate, or perhaps to adjudicate all cases. But I would propose to this group that the threshold of algorithm accuracy considered fit for purpose really is unclear. And in my role as an editor at Pharmacopoeia and Drug Safety, we get papers that come in 72% accuracy, 75% accuracy, fit for purpose, they say. Is it really? Several considerations after you have done your validation of a case finding algorithm. First, I often advocate sensitivity analyses, sensitivity analyses, sensitivity analyses. Using various definitions of the algorithm is often very valuable to determine the robustness of the results. That number one. Number two, I alluded to the fact that I get questions. I validated in one data source. I want to use it in another. Highlight to you, algorithms are usually not transportable. Different populations, different disease prevalence, different coding, different practices. Accuracy may differ by database. You may want to consider revalidating in the new database. Lastly, this process of conventional validation, typically single validity measure, most often positive predictive value. However, just looking at positive predictive value may mask differences in algorithm accuracy by exposure group. If the PPV or the sensitivity differs for that algorithm in the exposure groups, misclassification can actually be quite large. So what do we often do with these validity indices after the validation study? How do we use them to show if or how misclassification affects study findings? In some papers, investigators have used statistical methods to correct the measures of effect in studies for imperfect algorithm accuracy. In a classic paper, Brenner and Geffler use only PPV to correct a measure of effect. However, I would highlight to you, this assumes non-differential sensitivity. And I just said to you, algorithm accuracy may differ by exposure group. Rosa and the ISPI database SIG, Gillian Hall, developed an app that uses a range of plausible PPVs and sensitivities in the exposure groups to correct effect measures. Dr. Schultz is going to talk to us about quantitative bias analysis. So another option is to use quantitative bias analysis to determine the magnitude of the bias of the association due to algorithm inaccuracy. Very valuable approach. But we typically need a range of algorithm sensitivity and specificity in the exposure groups. And I just said that for most, we just use a single measure of, a single validity index. What we heard from now Dr. Lemoncello's PhD thesis yesterday is potentially using a high sensitivity screening algorithm that was initially proposed in PDS by Lanes and Beecher, and he has taken it to the next step. I'm going to let him talk about this sensitivity screening algorithm as a way to identify the sensitivity of an algorithm in each exposure group. But Bellertz and Genie, who sits right next to me, also had done a nice paper that if you have one validity measure and the disease prevalence, or you have two validity measures, you can derive the other validity measures, which will be very valuable for quantitative bias analysis. And then lastly, I just want to highlight to you, increasingly we are using AI to identify health conditions, machine learning, natural language processing, and our group has been doing this for liver diseases using deep learning approaches to measure fat in the liver and natural language approaches, natural language processing approaches at right to identify hepatic steatosis. So to summarize, I think I've given you some sense that algorithms for health conditions may be inaccurate, that these validation studies really are valuable to assess the potential for algorithm misclassification. I've reviewed with you the important steps in conventional algorithm validation, how we've talked about you can use the validation indices to inform quantitative bias analysis, and we've talked about the future and AI. And so I conclude with two considerations for Zabi and Ercilia and Robert. So considerations for Xabi during protocol development. Xabi, I would say you should consider assembling experts in methods, statistics, data, clinicians to inform the design of your validation study and your protocol. All too frequently, I see a disconnect and siloing. And what I think makes a more robust protocol is bringing disparate groups together around the table. I think it's important to include validation of key variables, whether it be eligibility criteria, outcome, either, if not previously done. Consider using the validity indices of the outcome to inform quantitative bias analysis, and potentially in the future, in your protocol or in the future, consider AI to aid abstraction, reduce measurement error. Methodological questions I have for Ercilia and Robert. What threshold of algorithm accuracy should we as a field consider as fit for purpose? This has relevance because we need to sort of know how low should we say, we may need to refine that algorithm or completely abandon it. Should validation of health outcomes routinely be performed by exposure group to assess for differential misclassification? I hinted to the fact that the accuracy may differ by exposure group, not routinely done. What should be the preferred approach to address algorithm inaccuracy after the validation study is done? Should we statistically correct our effect measures based on these indices? Should we use quantitative bias analysis, or both? So I thank you for the invitation to speak today to all of you. I thank you for the opportunity to come to Firenze. It's beautiful. And I thank you for your attention from the city of brotherly love, Philadelphia, Pennsylvania.
Thank you so much, Vin.